Making a human protein using biotechnology, growth in bacteria basically starts with the bacteria. This is a strain of bacteria that's really designed to make a lot of protein and not degrade it. And so we basically design and synthesize a circular plasmid, DNA, that we heat shock into a bacteria strain. It's kept stocked in the deep freeze at -80, and every couple of weeks we streak some out onto these growth plates, and we streak them out till we get separate colonies of growth. That is scraped off a plate with a loop that we can flame red hot to sterilize it. It can be cooled in the media. You can scrape some of the cells off with this and put it into some media. In this case today, it's going into a overnight starter culture. It's a small flask growing in here at 37 degrees. It's a LB, that's a Luria broth. It's got a little extra glucose in it for good growth, and there's ampicillin put in it, and just the antibiotic ampicillin. The plasmid that's in the bacteria that makes the RNA to make the protein we want is ampicillin resistance is on the plasmid. So by having this plate and the growth media with ampicillin in the media, that makes sure that all the bacteria as it grows in the culture will keep the plasmid, because we'll need that to express protein. Then this will grow overnight, and that starter culture will be used to start a larger amount of culture, say maybe 100 or 200 mils in another flask, and that will grow. We'll add a chemical called IPTG to induce growth of the protein, and we grow it for a few hours and collect the bacteria, and that's going to be a source of milligram quantities of human protein if you scale it up to just a few liters. And that's how we make proteins that we're developing for future therapeutics for retinal vascular disease.